This is the BioCentury Show. Brought to you by the inaugural BioCentury Grand Rounds Europe, debuting in Cambridge, UK this September. Connect with VCs, academic innovators, and biopharma R&D and BD leaders to explore groundbreaking advancements in early-stage R&D and dive into disease biology, therapeutic modalities, and enabling technologies. Learn more at biocenturygrandroundseurope.com.
Hello and welcome to the BioCentury Show. I'm Simone Fishburn, Editor-in-Chief at BioCentury. It is really a delight to be joined today by John Lepore, CEO of Profound Therapeutics and a CEO partner at Flagship Pioneering. Previously to that, John was SVP and Head of Research at GSK, where he spent 17 years in all. John is a cardiologist and internist by training and was a faculty member in cardiovascular medicine at Penn early in his career. And maybe your crowning credit, John, is that you are a trusted and valued member of BioCentury's Scientific Advisory Board.
Absolutely.
So to all of those accomplishments, I'm sure, the crowning achievement is that certainly it is for us, a very valued member. So welcome. I do want to start with your journey from GSK to being an independent CEO. Just talk a little bit about the driver for making that move. I want to point out that there's been quite a few people who've left the higher ranks of pharma and many of your peers, right? And they too have gone on to either create new codes or head up companies as CEO. And maybe you can talk about the driver for you in that move.
Yeah, super. Well, first of all, thank you so much to you, Simone, for having me and for hosting this, terrific forum. Being part of your SAB has been like a tremendous learning experience for me as well. So I'm happy to engage further here. Yeah, look, everyone's got their own career sort of trajectory and interests and goals. It's interesting, as you point out, that there's a confluence of a fair number of people who've gone in some version of the direction I have, which is from big pharma to small company. I'm pretty sure I can't speak for them, but I can give you my own example and sort of a few points that illustrate how I got there. First of all, for me, underlying all of these activities I've been involved in is just a personal passion about how can I best leverage the science that's been emerging over those several decades to help patients in the most sort of profound way. And it is a play of words, and that's one of the reasons I really liked the idea of what Profound is doing. But for me, having a profound impact started with like literally seeing patients on a one-by-one basis, which was enormously enriching personally. I hope I helped some patients as well. But I realized that that takes a certain type of intellect and person to do that for many decades, as so many of my mentors had done. But it wasn't the right thing for me. And I became enamored with how can I be involved in sort of helping mature science and then applying it at greater scale to other people who are going to, impart it one by one to patients. And that's how I got into being involved in the lab, involved in academic medicine, translational medicine. Then for me, it was a transition to, okay, well, wait a minute, I might be able to have a bigger impact doing translational medicine in a large pharma company, which by my definition, that's all they do is translational medicine. They're there to take science, make pills or antibodies or something, put in people, translate the science into real efficacy, and then bring it to patients in the market. So I was very excited about that. Had a tremendous run at GSK, which I've got enormous respect for and continued sort of connectivity to. But then, this idea came along to look at Profound Therapeutics. And I know we're going to get into this maybe a little bit later, but for me, the science was so compelling with my expertise and sort of career sort of tools that I thought I could just make the biggest impact for patients. And I made a leap and here we are.
So, one of the reasons I ask this, and it's going to relate to my next question, is that, I tend to sometimes take a step back and look at the industry as a whole and the ecosystem. And I think it's very clear that pharma companies are different than they were 20 years ago and even 10 years ago. I think, and I've written about this, we have various forces changing. I think, when we come out of this era, we're going to look different as an industry. But biotech companies, there's been a lot of externalization of science. Now taking the same talents that would have been doing what they were doing and able to do that inside pharma companies. As I said, a lot of people reach the top and then they're like, actually, I want to go to a smaller company where I can, leverage my skills differently. But what we've now got going on is a very capital constrained environment. There's generally a feeling that too many biotechs were created. I don't know if there's a feeling that there's the right number of pharmas. We won't get into that right now. But you being Profound, recently announced a deal with Novartis. And I think you have another one coming soon. And last year you announced one with Pfizer. So I wonder if you can talk, especially to those people out there who are CEOs who maybe haven't been inside pharmas, talk about deal making in this era. How much pressure is there in this capital constrained environment to create deals for revenue, to actually bring in some upfront money? Yeah. And, how much competition was there out there?
Yeah, I mean, there's a lot of things around that question and the present environment. But I think there's a few key points that come together where there's alignment between what's going on in the pharma space and what's going on in the biotech space that's really enabling these kinds of collaborations. So on the pharma side, there's equally financial pressures. But there's also, in essentially all pharma, near-term to deliver on the proximal pipeline. And when you have that combination of we've got to make the marketed products and the development products work and our budgets being pushed, there's downward pressure on the research budget. That's an environment where pharma R&D organizations are saying, let's find targets, projects, molecules that are a little bit further advanced that I could access them through a BD partnership without having to employ as big of an internal group. So that dynamic is driving their interest in collaborations with companies like ours.
Well, a couple of things there. So to the point that you just made, what these, at least the disclosed deals are about is not mature assets, but actually research partnerships, right? But the other part of my question actually is that it's very clear from your answer, you've sat both sides of the table. You really understand, their drivers. And so how much does that, can you answer the first part, but also how much does that inside knowledge really inform your conversation?
Well, on the second part, it's been absolutely invaluable. I'm fortunate to have lived a little bit in both worlds. It helps to know why the other party is asking the question. It helps to know how your answer is going to be interpreted.
Right.
And it helps to know how to present things in a way that is attractive to them, not to oversell it, but rather to put it in the terms that, they're making the decision on. So yeah, there's no doubt that helps a lot. But yeah, on your point though, yes, these are research phase collaborations, but they're not in completely new exploration. In the sense that what we've done in our company, maybe to segue into that a little bit, is over the past 4 years, really build up a capability and a platform that's identified an amazing new world of proteins. These are proteins that are not annotated in the known proteome. They're not the 20,000 that were identified in the Human Genome Project. Hopefully we'll have a chance to talk a little bit more about the science, but we've got a novel methodology for finding these. So it's not about searching. What we've done now is package them as very enticing drug targets because we've been able to put around them various features. For example, we've shown their human genetic association with disease. We've shown their expression in human tissues. We've started to do the biology that puts them in context of human disease. We've shown that they're tractable as drug targets. In some cases, we've raised antibodies against them. So for pharma, it's not sort of starting de novo with the research project. It's starting pretty far along with like tractable targets. And that's what there's a real paucity of. And that's why I think both Pfizer, Novartis, and as alluded to, we've signed a deal that we haven't announced yet, but I could sort of speak about it indirectly. These three companies have been very excited to access targets.
So let's just stay for a moment with the company strategy. And your platform is very interesting. As I said, we will get into that. But platform companies come in and go out of fashion. I feel like with the explosion of biology and technologies, it's really fertile ground for building platforms. But at the same time, the market and investors, they want products, right? So how much is that a tension for you regarding investing in the platform versus the pressure to come up with named products for investors or for raising money? And how are you managing that balance?
There's enormous pressure on that. And, we've had investors that literally have said, oh, do you have any, IND ready molecules? We're like, well, the whole point is I introduced this as, a target discovery company. So of course I know that's the point. I need your money to advance it to get to that. But their pressures are like find things that have high value. They could, monetize pretty quickly too. So, all the parties have sort of different pressures in this context. So for me, that platform conundrum is an and, it's not an or. So platform companies like ours, and I'm part of the flagship ecosystem, and this is generally the way we think about this. First of all, you don't want to build a platform for which there's no conceivable molecule or medicine that could come out of it. In fact, you don't even want to build a platform for which you think there is one or two. The point of a platform is to be a sustainable platform for sustained delivery of things of a new quality or a new opportunity or a new capability that could lead to multiple different medicines. If you spend all of your money and time up front on perfecting a platform that either can't get to more than one or two medicines or takes way too long to get there, that gap is very hard to bridge. And that's where I think a lot of platforms, they run out of money and they run out of energy and they run out of interest. The trick is to find one where you've got those front end features, fascinating science, novel approach, applicability across multiple different therapeutic areas, opportunity for multiple medicines, and very early on, you start to demonstrate that opportunity with specific programs. And we've been able to do that so far, particularly in oncology, but increasingly in metabolic and immune disease. And, having that and is, I think, what's going to be very important for our success.
And to what degree I'm coming back to the deal making now, because, I know that one of your deals looks at, I think, obesity targets, and I think another is cardiovascular. Yep. So to what degree does that partnership help focus you towards the specific products in mind, or is that just that they're joining the journey and it's not really changing how you would do things anyway?
No, it's brilliant because the reason we were so excited about partnering in those two cases with Pfizer and Novartis was because they've got such deep expertise in all aspects of bringing the molecules forward, creating the molecules, having the right translational models, having the infrastructure to do the clinical experiments, and the commercialization infrastructure as well. So the whole point is leveraging that expertise to bring our science to patients more quickly.
So it's not a simple handoff that you get it to a stage and they take it from there.
That would be unhelpful for both parties, really. Right? So it's really about the synergy of applying our science, their capability, and doing something that was, the proverbial 1 + 1 = 3. The other thing for us, Simone, strategically, because this platform is applicable across so many different areas, we would never have bandwidth to tackle all of the problems that could be solved with the platform. So by leveraging these pharma collaborations, we're getting done more for patients than we would have been able to do ourselves. And that's exactly where we want to be.
Well, I think that's a great place to take a break. And when we come back after this short break, we're going to actually dig into some of the science of Profound's platform. All right.
Super.
Grand Rounds, BioCentury's R&D conference, will make its European debut in 2025. Join us to debate key translational bottlenecks and unlock scientific breakthroughs with commercial potential. Connect with VCs, academic innovators, and biopharma R&D and BD leaders to explore cutting-edge advancements in early-stage R&D as we dive into disease biology, therapeutic modalities, and enabling technologies. Join us for the inaugural BioCentury Grand Rounds Europe in Cambridge, UK, the September 17th to 19th. Register and learn more at biocenturygrandroundseurope.com.
We are back at the BioCentury Show with John Lepore, CEO of Profound Therapeutics. So, John, we teased this a lot in the first half, but let's talk about the science of Profound's platform. I know, and a previous guest of mine, David Altshuler from Vertex, turned to what was behind him on his screen and brought that into the conversation. So I'm hoping that we're going to be able to bring what's behind you into the conversation. The premise of your platform is that there might be tens of thousands of proteins that others have missed, right? And walk us through that premise and where you're going with it, and tell us about that map.
Okay, will do. First of all, Dave Altshuler and I trained in medicine together many moons ago, so it's not surprising we have a confluence of maybe approaches or interests in some way. The map over my shoulder is a 1511 map by a German map maker named Waldseemüller. And there's one copy of it. It's in the U.S. Library of Congress. The U.S. government paid something like $15 million for it. Because it has a distinct feature. It's a little bit hard to see, but on the far left, it's the first time that the continents of North and South America were shown on a map, and it's the first time the word America appeared on a map. And the reason I have it here is we think about the discovery of novel proteins as being completely analogous to that. When the company was founded in 2020, if you looked in a textbook of biology, or if you asked any geneticist, how many proteins are there in the human body? They would have said somewhere around 20,000. And then you'd say, well, how do you know that? You say, well, there was something called the Human Genome Project that was done, back in the 90s, which was an outstanding feat for mankind. But it had some limitations. At the time, based on the technologies that were available, they applied a set of rules to determine what was a novel gene or a protein. And those rules ended up now, in hindsight, being fairly restrictive. For example, every protein had to be more than 100 amino acids. Well, insulin and glucagon are pretty good medicines, and there's 100 amino acids. And there are other rules in place. And that caused us to think, well, okay, maybe there might be other proteins if we relax the rules. And, from a pharma point of view, the context is that of the 20,000 proteins, somewhere around 600 have been targeted therapeutically with effect. And well, if there were 20,000 more, might there be 600 more drug targets? And, one of the challenges in pharma, I live this every day when I was running the research organization, was that as soon as you find a good target, you hear footsteps because every other company is finding the same thing because they have the same tools. So what we're doing is defining a novel pool of targets, a novel pool of proteins from which to identify new drug targets. And it's pretty cool. Fundamentally, there's a platform technology that allowed us to find these. And if you'd like, I could talk about that, or looks like you might have another list of questions.
No, I do want to go into that, but I just want to make the point that, so, in a way, it's a very fundamental question that you're addressing. It's sort of, what makes, let's say, an open reading frame an open reading frame? Is it 4 in a row? I mean, there are obviously, we used to discuss when is a polypeptide a protein, back in the days when I did my PhD, and so you're really sort of, eliminating those boundaries in a way, because I suppose you don't really care. What you care is whether it's an agent that's therapeutically targetable, if that's a word. So going back now, it seems, from your platform that there's a lot of omics, right? You are triangulating, let's say, omics of I think I read the word translateomics, if that is
Yeah,
an actual word.
translateomics. Yeah.
Translateomics, that's a bit of a stretch. Okay, but let's go with it. Translateomics with transcriptomics, obviously functional genomics. So what is the process? I mean, that seems sort of in this omics world. Break it down for the non-omics people about how you go from there to your targets.
Yeah, the inventive step is the translatomics part. So if you tried to, we were starting to go there, reason, like use rules to look at the genome to say what could be an open reading frame. There's different predictions, but I think it's somewhere around 7 million different predictions. Okay, then it becomes,
Is there a minimum for an open reading frame in this context?
again, you could define it in different ways, but we don't think of it that way as DNA sequence. The technology we have, the translatomics bit, is to identify in an open-ended way exactly which transcripts are in the process of being translated at the ribosome. So we have a way of capturing the RNA sequences that are being translated. And then there's computational methods that then sort of stitch all these pieces together. And what we do is in silico and through this experimental bit, identify a length of RNA that we know was in the process of being translated in the ribosome. And then we use mass spec to confirm that the protein actually exists.
So it's a wet stage that is reading the RNA as it's being created, and then an in silico stage that is combining that. Is that what you're saying?
Totally. Wet stage with a computational algorithm. And now we've done this so much, we've basically industrialized that process, and we studied more than 400 different human cells and tissues. And what that's done is allow us now to superimpose a machine learning layer where we could start now to have like a predictive loop. Like if we see this, it's more likely going to be a protein, more likely going to be seen by mass spec, and it's got a positive feedback loop. So we've got very good about identifying these proteins and their confirmation by mass spec. There are in the range of several tens of thousands that we're pretty sure exist. And they've got some fascinating features. For example, they have domains like transmembrane domains, signal peptides, binding domains, just like the annotated proteins. They have cell type specific regulation. They're up and down regulated in disease. And they have many of the physical chemical properties of the annotated proteome. So it's pretty cool what's out there.
So it sounds very cool. And, to some degree, this is a real unbiased approach, right? Now you put a lens on it that says, I want obesity targets or I want cardiovascular targets. How does that layer in then? How does that bias, I suppose, get introduced?
Yeah, so that's the second part of the platform. So the platform has three components. The first is what I've described so far, this translatomics identification and validation of proteins. The second then is to put them into a disease context. And to do that, what we've done is build a very large database. We call it our Profound Atlas, play on words with the maps, where what we've done is layered many omics data sets. Some we've generated, but many that we've brought in from external sources. And then because we knew the sequence of every protein from the first step, we could look in the database and know exactly where the mRNA is expressed. We could predict, for example, the structure with AlphaFold. We know what motifs it might have. And very importantly, we know, is the novel protein encoded by a locus that has a human genetic link to disease? And this is one of the most powerful things because we found now thousands of proteins where we're essentially deorphanizing GWAS variants that could not previously have been connected to a protein. And we now realize that variant actually is a coding mutation in the novel protein we identified. So this is like a really important new pool of targets.
So you mentioned machine learning. I'm going to kind of ask about that in a minute, but as I listen to you and you're building a company in 2023, I guess, to 2025 and so on, it's a very innovative company. using a lot of new technologies. So my question now is about your staffing and hiring.
Yes.
What are the skill sets that go into that you need? Do you need now data scientists? Do they actually need to know biology? How do you build a team at a staff level, not just your leadership, that can support this kind of research-based product development is what I should say, actually.
It's a super question and a well-timed one because the company you've been describing is in transition from a company that was essentially building a platform to now one's both building a platform and creating a drug discovery pipeline. So, we've had to augment who were the initial team, who were sort of thoughtful, creative scientists, platform builders, understood computational methods, and all the science of transcription and translation. We need that to continue to enrich the platform, but now we're in the process of superimposing people who've got drug discovery and development abilities that can take those proteins and really put them into actual programs to de-risk the targets.
So I'm going to go from there to AI and machine learning. And I think I'm just going to ask this question. To what degree across the company is AI a part of how you are building this biotech?
Yeah, it's becoming an increasingly critical step because at the beginning we needed to build the mechanics of the company, how to get this done, how to find the proteins. And as your listenership will well know, to properly use machine learning and AI, there's a necessity to have a very large data set that's extremely well curated. So we spent the first couple of years of the company building that data set. So we've got the largest, by far, compendium of these novel proteins and their context in human disease, as I described. What we need to do now, and what we are doing, is building really essentially agentic AI models that could start to link these novel proteins with what's known about the annotated proteins. So you could picture, if I had a figure here, I would have.
So tell me the agentic part of this. Like you teach them and then they build it for you.
Exactly. Yeah. So if you can imagine a protein network, dots of connectivity of all the 20,000 known proteins. A lot of people have that. Okay. Right. What we're doing is filling in blanks of other proteins that actually exist in that network and they weren't known before.
So this is like relationship sort of
relationship.
Right.
Yeah. So we don't want to look at our proteins, in themselves, but rather how do they relate to what's already known about the biology of disease and the other proteins? And then you're instantly informed about which ones of our novel ones are more likely to have an important therapeutic role in disease. So that's what we're building now. Very exciting period. You need experts in that. And we just hired a new CTO, Tom Chittenden, who's just joined, and we'll be building that capability as well.
Oh, that is very exciting. I do want to talk about in the last few minutes about the innovation environment. And actually, I am going to start with a question about China. And I could have asked it earlier, but I felt that it really is, for me, part of the innovation landscape. And our analysis, BioCentury's analysis, which will, it shows that a very large proportion of deals with China biotechs, or Asia biotechs really, are driven by biotechs in the West as opposed to pharmas. Actually, there's more. Now, at the same time, I've seen a lot of Western biotechs alarmed by the rising competition in China. So first of all, I sort of want to ask how you are thinking about that landscape and harnessing or partnering or watching what's going on among China biotechs.
Yeah, I mean, it is really fascinating to watch. I think, there's a lot of factors to consider, but net, building the world's capability to make novel medicines quickly and move them quickly is probably good for patients. So, where that works, understatement of the year, I welcome the opportunity to do things quickly. Now, for us, as I said, we're on the front end of starting new programs. We do a lot of discovery work, like CRO-related work in China, like many people do. Most of our programs initially, we're partnering with large pharma. Now, some of them in turn have collaborations with China. Others have internal capability. And, I think we'll let this play out. I mean, there's
So,
optionality. I think China's providing optionality to do things quickly.
yeah, I think that's a great way of looking at it. I do want to talk more domestically in the US,
Yeah.
where NIH funding cuts have got people very worried about research in general. And I realize that this is a feature that is important for investments, for VCs, where this is where a lot of the innovation is coming from. But how much, if at all, does that affect a company like yours?
Look, I think potentially enormously. The U.S. government funding of science has been fundamental to advancing scientific endeavor worldwide. And it was a quantum change, starting from World War II-ish through now. So, pulling that out precipitously, as some of the discussion has been about, will have a lasting impact for everyone, including us, because all the tools that we're ultimately using somehow were enabled by U.S. federal government funding. So we're building on Human Genome Project. Okay, who funded that in part at least? All the technologies for AI ML somehow have been incubated at different academic institutions that have, like, we could go right down the line. So, a world where that funding in a lasting way is not present, I think is going to slow us all down.
Well, I'd like to end on a positive note. So we will see you at our Grand Rounds conference in Cambridge, UK, where we're going to dig into the academia industry interface. And I think that what you're suggesting, and maybe you want to have the last word, is that this is a very fertile interface. There's really a lot going on in academia that really is the platform and foundation and supports what's going on in industry.
Absolutely. Look, these dynamics of how small companies, large companies, academic institutions, and government-funded institutions work together is just it's our own protein network that keeps the organism going. And, I'm just thrilled to have the opportunity to be working in an area of science in a particular company that has opportunity to interdigitate with all of that, and really psyched about what we specifically as a company could do for patients, but more broadly with the whole enterprise, despite all the noise, will continue to do. And looking forward to talking more about it in London with you and the team.
Well, thanks, John. We look forward to seeing Profound's progress. We will keep an eye out for your next deal announcement. And thank you very much.
Thank you.
Brought to you by the inaugural BioCentury Grand Rounds Europe, debuting in Cambridge, UK this September. Connect with VCs, academic innovators, and biopharma R&D and BD leaders to explore groundbreaking advancements in early-stage R&D and dive into disease biology, therapeutic modalities, and enabling technologies. Learn more at biocenturygrandroundseurope.com.